Hello and welcome, dear students. Today we are going to discuss about the topic human ape comparisons. The main objectives of today's lecture are to understand the basic scenario of human ape comparisons, to know the historical perspective of human ape comparisons, to discuss the emotional expression in human and non-human great apes, to discuss the bipedalism in humans and apes, to understand the sexual dimorphism in humans and apes, to discuss the karyotype of humans and apes, and finally to know the evolutionary relationship of primates. Dear students, let's start with the introduction first. It has been historically difficult for people to accept that humans are in fact just another primate species with African origins and that humans differ physically only in degree from some of the other. The similarities can be seen throughout our bodies. The African apes and humans have essentially the same arrangement of internal organs, share all of the same bones, though somewhat different in shape and size, lack external tails, and have several important blood type systems in common. Humans also get many of the same diseases. Humans and African apes have hands with thumbs that are sufficiently separate from the other fingers to allow them to be opposable for precision grips. Like all of the great apes, humans are sexually dimorphic. Human men are 5 to 10% larger on average and have greater upper body muscular development. Like chimpanzees and bonobos, humans are omnivores in nature. The comparatively minor anatomical differences between humans and apes are largely a result of our habitual bipedalism. A number of changes in human bodies were related to the evolution of this form of locomotion. Unlike apes, human arms are relatively short and weak compared to legs. Our feet no longer have the ability to effectively grasp and manipulate objects because the toes became shorter and the big toe moved up into line with the others. The human pelvis and spinal column also have been modified for an erect posture and efficient bipedal locomotion. Research done by Herman Pontzer of Washington University in St. Louis indicates that longer legs require less up and down movement while running and therefore reduce the amount of energy needed to move rapidly. In addition, the largely hairless human body with its abundance of sweat glands allows us to remain cooler while running than if we only relied on panting like most other mammals. This, no doubt, was a major advantage for our early human ancestors in the competition with other hunters and scavengers for meat in warm climates. Dear students, a partial evolutionary solution to birth difficulty for humans was fetuses being born at a less mature stage when their heads and torsos are smaller. The trade-off is that human infants are more vulnerable. By comparison, chimpanzees at birth are neurologically and cognitively ahead of human babies of the same age. But the chimps begin to fall behind by about 6 months old because of the more rapid continued development of the human brain following birth. With the exception of these differences, humans are quite similar to the African apes anatomically and genetically, especially to the chimpanzees and bonobos. Humans have 46 chromosomes in their cells while all of the great apes have 48. In reality, this difference is not as great as it would initially seem because the human chromosome 2 is a fusion of ape chromosome 12 and 13 with most of the same genes. Research on learning the entire genome of common chimpanzees was completed in 2005. A comparison between this and the human genome completed in 2001 shows that 96% of DNA-based paired sequences of humans and chimpanzees are the same. Most of the 4% differences is in duplicated non-gene segments. If only gene segments are compared, there is a 98% similarity. The genes that differ mostly control speech, smelling, hearing, digesting proteins, and susceptibility to certain diseases. These dissimilarities are to be expected given that humans have been on essentially separate evolutionary track for 6 to 7 million years. The modern human brain is 3 times larger in volume than those of the great apes. More importantly, the human brain to body size ratio is significantly larger, and it has a much bigger cerebral cortex with a higher concentration of neurons. The human brain uses about 25% of the energy derived from the nutrients that we consume and 20% of the oxygen. Recent research has suggested that our intelligence advantage may be due to evolutionary change in the HAR1F regulatory gene beginning about 6 million years ago in our pre-human ancestors, but not in those of chimpanzees or other apes. This gene is involved in the production of brain tissue between the 7th and 19th week after conception. It's not surprising that there are some striking differences between the great apes and humans in mental abilities. People have much more complex forms of verbal communication than any other primate species. Humans are the only animals to create and use symbols as a means of communication. The great apes are also surprisingly intelligent, having mental levels equivalent to 3 to 4 years old human child. This is sufficient to allow them to learn and use the sign language of deaf humans in at least a rudimentary way, but they do not have the capacity or capability of producing human speech and language. This is likely due to the fact that they have a different form of another key regulator gene known as FOXP2. Dear students, let's understand the historical perspective of human-ape comparisons. The divergence of humans and ape ancestors dates back to approximately 6.5 to 7.5 million years ago or even earlier. It is still of a great interest to identify genetic elements that distinguish humans from apes and encode features of human physiological and mental identities. It is a difficult task to quantitate the exact percentage of differences between human and ape genomes. In early works, divergence of human and chimpanzee genomes was estimated as roughly 1%. This estimate was based on the comparison of protein coding sequences and did not consider non-coding, that is, major part of DNA. However, the idea of 99% similarity of genomes persisted for a long time until 2005, when nearly complete initial sequencing results of both human and chimpanzee genomes became available. It was found that genome differences represented by single nucleotide alterations formed 1.23% of human DNA, whereas larger deletions and insertions constituted 3% of human genome. Moreover, even higher proportion was shaped by chromosomal inversions and translocations comprising several megabase long chromosomal regions or even entire chromosomes, as for the chromosomal fusion that took place when the human chromosome 2 was formed. Dear students, let's now discuss about the emotional expression in human and non-human great apes. Emotional expressions are frequently used in all great ape species. Compared to apes, humans have evolved communicative faces where all the expressive parts are emphasized. Also, in great apes, species-specific facial characteristics have evolved to enhance communication. Smiles have been ritualized to a greater extent in humans than in apes. Laughter is similar and seen in comparable contexts. Great apes do have voluntary control over some expressions. In chimpanzees and bonobos, facial and vocal displays may be more narrowly context bound than gestures, but whether this extends to bodily expressions of emotion and how that compares with humans is not known till date. While progress has been made, many more studies of emotion production are needed to enhance the understanding of the mechanism of emotion expression in humans and apes. Recent technological advances, including thermography, papillometry, can improve insight into the underlying mechanism invisible to the naked eye. Dear students, let's now discuss about the bipedalism in humans and apes. Bipedalism is the basic adaptation of the hominid and is considered the main cause behind a suite of skeletal changes shared by all bipedal hominids. The earliest hominin, or presumably primitive bipedalism, is considered to be either Sahelanthropus or Ororin, both of which arose some 6 to 7 million years ago. The non-bipedal knuckle walkers, the gorillas and chimpanzees, diverged from the hominin line over a period covering the same time, so either Sahelanthropus or Ororin may be our last shared ancestor. Ardipithecus, a full biped, arose approximately 5.6 million years ago. The early bipeds eventually evolved into the Australopithecines and still later into the genus Homo. There are several theories of the adaptation value of bipedalism. It is possible that bipedalism was favored because it freed the hands for reaching and carrying food, saved energy during locomotion, enabled long distance running and hunting, provided an enhanced field of vision, and helped avoid hyperthermia by reducing the surface area exposed to direct sun. A study provides support for the hypothesis that walking on two legs or bipedalism evolved because it used less energy than quadrupedal knuckle walking. Another change is the shape of the big toe. Recent studies suggest that Australopithecines still lived part of the time in trees as a result of maintaining a grasping big toe. This was progressively lost in habilines. Anatomically, the evolution of bipedalism has been accompanied by a large number of skeletal changes, not just to the legs and pelvis, but also to the vertebral column, feet and ankles and skull. The most significant change occurred in the pelvic region, where the long downward facing iliac blade was shortened and widened as a requirement for keeping the center of gravity stable while walking. Bipedal hominids have a shorter but broader bow-like pelvis due to this. A drawback is that the birth canal of bipedal apes is smaller than in knuckle-walking apes, though there has been a widening of it in comparison to that of Australopithecine and modern humans, thus permitting the passage of newborns due to the increase in cranial size or capacity. This is limited to the upper portion, since further increase can hinder normal bipedal locomotion. The shortening of the pelvis and smaller birth canal evolved as a requirement for bipedalism and had significant effects on the process of human birth, which is much more difficult in modern humans than in other primates. The smaller birth canal became a limiting factor to brain size increase in early humans. The increased brain growth after birth and the increased dependency of children on mothers had a major effect upon the female reproductive cycle and the more frequent appearance of alloparenting in humans when compared with other hominids. Dear students, let's talk about the encephalization. The human species eventually developed a much larger brain than that of other primates, typically 1330 cm per cubic, that is 81 cu in modern humans. Nearly 3 times the size of a chimpanzee or gorilla brain. After a period of stasis with Australopithecus, Anaminensis, and Ardipithecus species which had smaller brains as a result of their bipedal locomotion. The pattern of encephalization started with Homo habilis, whose 600 cubic centimeter brain was slightly larger than that of chimpanzees. This evolution continued in Homo erectus with 800 to 1100 cubic centimeter and reached a maximum in Neanderthals with 1200 to 1900 cubic centimeter, larger even than modern Homo sapiens. The brain increase manifested during postnatal brain growth far exceeding that of other apes. It also allowed for extended periods of social learning and language acquisition in juvenile humans beginning as much as 2 million years ago. The size and shape of skull changed over time. The leftmost and largest is a replica of a modern human skull. The temporal lobes, which contain centers for language processing, have increased disproportionately, as has the prefrontal cortex, which has been related to complex decision making and moderating social behavior. Other factors are the development of cooking, and it has been proposed that intelligence increased as a response to an increased necessity for solving social problems as human society became more complex. Change in skull morphology, such as smaller mandibles and mandible muscle attachments, allowed more room for the brain to grow. The increase in volume of the neocortex also included a rapid increase in size of the cerebellum. Its function has traditionally been associated with balance and fine motor control, but more recently with speech and cognition. The great apes, including hominids, had a more pronounced cerebellum relative to the neocortex than other primates. It has been suggested that because of its function of sensory motor control and learning complex muscular actions, the cerebellum may have underpinned human technological adaptations, including the preconditions of speech. The immediate survival advantage of encephalization is difficult to discern, as the major brain changes from Homo erectus to Homo heidelbergensis were not accompanied by major changes in technology. It has been suggested that the changes were mainly social and behavioral, including increased empathic abilities, increases in size of social groups, and increased behavioral plasticity. Humans are unique in the ability to acquire information through social transmission and adapt that information than other apes. The emerging field of cultural evolution studies human sociocultural change from an evolutionary perspective. Dear students, let's now understand the sexual dimorphism. The reduced degree of sexual dimorphism in humans is visible primarily in the reduction of the male canine tooth relative to other ape species except gibbons, and reduced brow ridges and general robustness of males. Another important physiological change related to sexuality in humans was the evolution of hidden estrus. humans are the only hominids in which the female is fertile year-round and in which no special signals of fertility are produced by the body, such as genital swelling or overt change in proceptivity during estrus. Nonetheless, humans retain a degree of sexual dimorphism in the distribution of body hair and subcutaneous fat, and in the overall size, males being around 15% larger than females. These changes taken together have been interpreted as a result of an increased emphasis on peer bonding as a possible solution to the requirement for increased parental investment due to the prolonged infancy of offspring. Dear students, let's now talk about the ulnar apposition. The ulnar apposition, the contact between the thumb and the tip of the little finger of the same hand, is unique to the genus Homo, including Neanderthals and anatomically modern humans. While as in apes the thumb is short and unable to touch the little finger, the ulnar apposition facilitates the precision grip and power grip of the human hand, underlying all the skilled manipulations. Dear students, let's now discuss about the karyotype of humans and apes. Human karyotype is represented by 46 chromosomes, whereas chimpanzee has 48 chromosomes. In general, both karyotypes are very similar. However, there is a major difference corresponding to the human chromosome 2. It has originated due to a fusion of two ancestral acrocentric chromosomes corresponding to chromosomes 2A and 2B in chimpanzees. Also significant, pericentric inversion were found in 9 other chromosomes. 2 out of 9 are thought to occur in human chromosome 1 and 18, and the other 7 in chimpanzee chromosome 4, 5, 9, 12, 15, 16, and 17. In addition, there are numerous differences in the chromosomal organization of pericentric, paracentric, introcalary, and Y-type heterochromatin. For example, the chimpanzees have large additional telomeric heterochromatin region on chromosome 18. Additionally, the majority of chimpanzees' chromosomes contain subterminal constitutive heterochromatin, that is C-band blocks, SCBs, that are absent in human chromosome. SCBs predominantly consist of the subterminal satellite, that is ST-SAT repeats. They are found in African great apes but not in humans. The presence of such SCBs affects chimpanzees' chromosomes behavior during meiosis, causing persistent subtelometric associations between homologous and non-homologous chromosomes. As a result of homologous and ectopic recombinations, chimpanzees demonstrate greater chromatin variability in their subtelometric regions. Studying sex chromosomes also revealed several peculiar traits or characters. There are several regions of homology between X and Y chromosome, so-called pseudo-autosomal regions or PARs, most probably arisen due to translocation of DNA from X to Y chromosome. The term pseudo-autosomal means that they can act as autosomes being involved in recombination between X and Y chromosomes. Both translocation and inversion took place already after separation of human and chimpanzee ancestors. Dear students, let's discuss the other changes. A number of other changes have also characterized the differences between humans and apes, among them an increased reliance on vision rather than smell, highly reduced olfactory bulb, a longer juvenile development period and higher infant dependency, a smaller gut and smaller misaligned teeth, faster basal metabolism, loss of body hair, evolution of sweat glands, a change in the shape of the dental arcade from U-shaped to parabolic, development of a chin, styloid processes, and a descended larynx are prominent in humans. Dear students, let's discuss the evolutionary relationship of primates. A comparison of DNA nucleotide sequence of living primate species shows that humans are most closely related to the African apes. Next, in descending order of genetic closeness to humans comes the Asian apes. Old World monkeys, New World monkeys, tarsaries, and finally the lemurs and lorises. This genetic comparison corresponds exactly within a comparison of homologous primate physical traits. It also fits nicely with what is known from the fossil record. The procimans were the first to evolve. Next came the monkeys, then the apes, and finally humans. Dear students, let's now conclude the lecture. Apes are humans' closest living relatives, and furthermore, apes and humans have more in common than most other animals due to the fact that we had common ancestors millions of years ago. This is the reason why humans and apes have features that are quite similar. Humans and great apes are highly social species and encounter conspecifics throughout their daily lives. During social interactions, they exchange information about their emotional states via expressions through different modalities, including the face, body, and voices. Dear students, it was all about today's lecture regarding human ape comparisons. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.